Greetings to everyone. I am Radheshyam Thakur and I am going to present on hydrophilic interaction chromatography. So let's first understand what do you mean by hydrophilic? Hydrophilic refers to material or surfaces that attracts water, absorb water, dissolve in water or have affinity for water. And as we all have dived into the concepts of chromatography, we all know that chromatography is a laboratory technique used to separate, identify, and quantify the components of a mixture. It involves passing a sample through a stationary phase which separates the component based on their interaction with the phases. In this presentation, we are going to learn about hydrophilic interaction chromatography. So let's first get to the definition of this. So what do we mean by hydrophilic interaction chromatography? As the definition says, hydrophilic interaction chromatography is a chromatography technique used to separate and analyze polar compounds that are poorly retained on traditional reversed phase column. It is particularly effective for highly hydrophilic substances such as amino acids, peptides, sugars, and nucleotides. There is somewhat similarity and differences between hydrophilic interaction chromatography and reversed phase chromatography. We are going to dive much more into detail in the latter part. Here we can see there is a beta cyclodextrin and there is hydrophilic interactions which is actually an ionic interaction and and um, and there will be other interactions as well which we will learn in the latter part. Going to the next slide, let us see what are some common examples of hydrophilic interaction chromatography. We all know that in this highly digitalized world that sugar is in high amount is very harmful for our health, and there are so many health conscious people who would like to know what are the sugar content in their food and beverages. So how do we actually get to know this sugar content? So the consumers who are often concerned about the sugar content in their diet, the HILIC helps food manufacturer and regulatory agencies monitor these levels. There are so many people who go to gym regularly and they require the right amount of supplement, vitamins, so that their recovery is much better. So the HILIC helps us to ensure that supplements contains the amount of vitamin they claim, providing safety and efficacy for consumers. And in the in the medicine world, analyzing the purity of polar drugs like certain antibiotics or blood pressure medications is very essential. And HILIC helps in these. Moving on to the next slide, here we will see hydrophilic interaction chromatography is a variant of liquid chromatography. It is actually liquid chromatography and has some advanced phenomena going on that is designed to separate polar compounds. This technique works by leveraging the interaction between hydrophilic analytes and a polar stationary phase. It is often described as a hybrid of normal phase chromatography but uses elements similar to reversed phase chromatography, especially in terms of mobile phase composition. HILIC is particularly important for analyzing compounds that are too polar to be effectively separated using traditional reversed phase chromatography. However, the key here is its focus on highly polar substance. This is crucial for fields like pharmaceutical analysis, where testing the purity of polar drugs is very essential. In the next slide, we are seeing that there is a there is this initial condition of biphasic solvent at silica interface. Initially, there is a homogeneous binary mobile phase high in organics content. It is well mixed and there is no separation between components. In the HILIC condition, we see that this biphasic system is actually divided into two parts: aqueous rich solvent and aqueous depleted solvent. The aqueous rich solvent is actually prepared when there is interaction between the SiOH and water molecules. Actually, the SiOH molecules binds the water molecule together and it creates an aqueous rich solvent. And there is this other system where there is aqueous depleted solvent, which means that there is less amount of water in that part. So actually this happens when a polar phase is utilized with a mobile phase high in organic concentration. The more polar water will preferentially absorbed on the surface, creating a semi-stagnant water in stationary phase. A water depleted mobile phase, polar and lattice then can into aqueous in which phase. When we go more into the details, is it that simple? Is there only polar interactions going on? Actually not. There are various other interactions also going on. Actually, the polar analytes are just that because they are ionic or exhibit strong dipoles. They are actually naturally interactive. We are bringing them close to a polar surface and therefore should expect other strong interactions to take place. These strong interactions can be strong dipole interactions, hydrogen bonding, and ion exchange, which are an integral and important part of HILIC chromatography. Here we can see that normetanephrine, actually here there will be hydrogen bonding also, because hydrogen is connected to an electronegative oxygen atom and nitrogen atom. There will be dipole interactions also, and there will be ion exchange also. So as discussed earlier, let us see the interactions in HILIC chromatography. There will be mainly three types of interactions and other interactions as well. First, the partition, where phase transfer of analytes to and from stationary phase take place. Polar interactions, which are actually dipole, hydrogen bonding, and induced dipole. Ionic interactions, where ion exchange with bonded phase or ionized surface silanol group take place. The SiOH was actually the silanol group here. There is one major difference between reversed phase and HILIC is the relative importance of this mechanism. In reversed phase, partition dominates and selectivity is generally tweaked by supporting secondary polar or ionic interactions. In HILIC, polar and ionic interactions often dominates. Considering this, when we see the difference between hydrophilic interaction chromatography and reversed phase chromatography, in hydrophilic interaction chromatography, as we have seen, stationary phase is the polar material. Mobile phase are organic rich solvents. In reversed phase chromatography, the stationary phase are the hydrophobic non-polar material. The mobile phase are water rich with organic solvent. The separation mechanism in HILIC chromatography would obviously be hydrophilic interactions, whereas in reversed phase chromatography it would be hydrophobic interactions. So in HILIC, the most hydrophilic compounds elute first and is suitable for polar charged or hydrophilic compounds. In reversed phase chromatography, most hydrophobic compounds elute first and is suitable for non-polar hydrophobic compounds. Moving on to the next slide. Here we can see there is two pictures of retention profile of cyanephrine on bare silica and pentafluorophenyl phases. The acetonitrile is the buffer diluted as organic is added. Actually, acetonitrile is the buffer phase. So what is buffer phase and what's its importance? Buffer phase actually controls mobile phase pH and ionic strength, regulate hydrophilic interaction between analyte and stationary phase, enhances retention and selectivity, and maintains column stability and reproducibility. So what do we understand by these two pictures? Let's see. The previous example shows that there may be very different mechanism causing retention and selectivity in HILIC system. Like the bare silica phase exhibits partition not impacted by buffer concentration. The PFP phase exhibit ion exchange chromatography highly impacted by buffer interaction. Differing retention mechanism gives rise to alternate selectivity and retention. So actually, if the material is different, the mechanism would be different. The bare silica phase, where it mostly functions on partitions, which is interaction between mobile phase and stationary phase, and the PFP phase, which exhibit ion exchange interactions. Let's dive into the components of HILIC system. So, as like every other chromatography technique, there would be a stationary phase column. The HILIC columns are packed with polar materials like bare silica, amide, diol, or zwitterionic ligands. All these are either high in dipole or exhibit strong hydrogen bond. These materials create a water layer on their surface which facilitates hydrophilic interaction. The mobile phase is usually a mixture of acetonitrile 70 to 90% and water or aqueous buffer 10 to 30%. The high organic content promotes analyte retention. Detector, often coupled with mass spectrometry or UV detectors for analyzing eluted compounds. Mass spectrometry is preferred due to the high organic solvent content, which enhances ionization efficiency. Pumps and injectors. High performance liquid chromatography pumps are used to deliver the mobile phase at a constant flow rate, while an auto injector introduces the sample into the system. The working principle of HILIC. Stationary phase interaction. The column's polar stationary phase is coated with a thin water layer that attracts polar analytes. When the sample is injected, analytes interact with water layer through hydrogen bonding, dipole-dipole interaction, and ionic forces. Mobile phase elution. The mobile phase has a high content of organic solvent, typically acetonitrile, with a small percentage of water or buffer. The organic rich mobile phase has low polarity, encouraging analytes to partition into the water layer on stationary phase. Retention mechanism. The retention of analytes is governed by their polarity. Highly polar compounds are more strongly retained due to their interaction with the water layer, while less polar compounds elute more quickly. This mechanism is the reverse of what happens in the reversed phase chromatography. So what are the key factors affecting separation? The key factors that affects the separation are mobile phase composition, in which if we increase the water content, it enhances the retention time for polar compound, but can also increase peak broadening. Thus, balancing acetonitrile and water becomes very crucial. pH and buffers. Adjusting the pH of the mobile phase can impact the ionization state of analytes. Common buffer include ammonium acetate or ammonium formate, which are compatible with mass spectrometry. Column temperature. Increasing the temperature can improve the solubility of analytes and reduce viscosity, enhancing the separation efficiency. Optimizing HILIC conditions. Choose the right column. Different stationary phases like amide versus zwitterionic will affect separation based on the analytes. Like according to our needs, we will choose the stationary phase. Mobile phase adjustments. Start with a high organic content, 80% acetoNitrile, and adjust water content gradually to optimize retention. Flow rate. A typical flow rate is 0.5 to 1 ml per minute, but this can be adjusted based on column dimension and pressure limits. Advantages of HILIC: high selectivity for polar analytes, compatibility with mass spectrometry, improving sensitivity, effective separation of compounds that don't retain well in reversed phase columns. Now we are going to discuss some challenges of HILIC. Since there is longer equilibrium time between runs, this can slow down sample throughput. Like since there is so much of time required between two equilibration, the sample injection becomes very slow. Complex optimization is required to balance the mobile phase composition, and it is sensitive to small changes in experimental conditions which can affect reproducibility. And there are some common issues with HILIC, and their solution is included here, and like poor retention. For troubleshooting this problem of poor retention, increasing the water content or adjusting pH to enhance interaction helps. Peak tailing. Peak tailing is basically a phenomenon where the peak is not sharp but is adjusted to one of the side. So to troubleshoot this problem of peak tailing, optimize the buffer strength and pH. High back pressure. To troubleshoot this problem of high back pressure, ensure that the mobile phase is well filtered to avoid clogging. We will now see some of the real world applications of HILIC system. Like in pharmaceuticals, the drug purity is very much essential, and analyzing polar active pharmaceutical ingredient which will then bind with the receptor cell is important. So in pharmaceutical industry, to analyze the pharmaceutical ingredients which are also called APIs, it is very much widely used. In the food and beverages industry, monitoring sugar content in juices or vitamin levels in supplement. And in clinical research, profiling metabolites in disease biomarker discovery. Now coming to the summary of this whole presentation, we can say that hydrophilic interaction chromatography is a highly effective technique for analyzing polar compounds that are challenging to separate using other chromatographic method. Basically, it is used for polar compounds. It's especially valuable in pharmaceutical, food, testing, and metabolomics. Thank you. This was all about HILIC, hydrophilic interaction chromatography. Bye.